Hello friends, Ace Botany, next episode. In this episode, we'll be proceeding with the microbes in human welfare second part. So for convenience, I will share the screen so we can proceed. I think screen came, so we can proceed. So chapter 8, microbes in human welfare part 2. Main discussion topics, as usual, there will be an introduction. Then microbes in industrial products, because household products we done in last episode. Then fermented beverages, antibiotics, then different chemicals, bioactive molecules. Then as usual, 5 theory question, 5 NEET model question with the key. So we can proceed as an introduction. See, this episode, we'll be discussing about use of microbes in connection with the different industries in the world. And one by one, we can proceed. So first is microbes in industrial products, starting with the heading fermenters. And that itself is a diagram-based theory question, NEET model question. Now, what is meant by fermenters? This is NCERT diagram. See, fermenters means large vessels used for growing microbes in connection with the industrial scale, because in industry, huge amount is needed. So that much size should be there for vessels like fermentation, etc. So that vessels, huge vessels, they are referred to be fermenters. I think idea clear, 1 mark theory question, then NEET model diagram-based question. So I think first page is clear, so we can proceed to second page. Now, fermenters, vessels. Now, next is fermentation, or connected with it, fermented beverages. So for this, we should know what is meant by the term beverage. Beverage, we are familiar with that term, a common term. So a beverage is a liquid that is intended to be consumed by humans. Example includes water, milk, juice, soft drinks, tea, coffee, chocolate drinks, alcoholic drinks, etc. But it should be portable. Portable means safe to drink. That is how classically beverage has been defined. So once you got the idea of beverage, we can proceed to further, that is fermented beverages. See, fermentation is an ancient procedure. Whether they know the science, they don't know the science anyway, they are doing this. See, one of the main organisms used was yeast from time immemorial, very ancient, for the production of beverages like wine, beer, whiskey, brandy, rum, etc. The specific yeast used to wear, or commonly used to wear, Saccharomyces cerevisiae. Yeast also concerned with bread making. So brewer's yeast, baker's yeast, different names will be there. That is Saccharomyces cerevisiae. And remember, yeast is a unicellular organism. It is a eukaryote. That itself is a question, eukaryotic unicellular organism yeast. For fermenting, different items will be utilized, like malted cereals, fruit juices, ethanol production, fermentation is involved. Yeast is widely used. And depending on the type of the raw material, and the type of processing, which raw material we are using, which process we are using, even which microbe, etc. So with or without distillation, that is why the type of processing, different types of alcoholic drinks has been obtained. So we can change the product by these mechanisms. So without distillation, what we normally produce is wine and beer, where alcohol content will be less. With the distillation, whiskey, brandy, and rum. So alcohol content will be more. That is how, depending on the treatment, we can make different products. For ethyl alcohol production, materials used include, materials, I think, one A is missing, materials used include molasses, maybe sugar industry waste, fruit juices, malted cereals by fermentation. The common one is molasses. Alcohol percentage can be increased by distillation. Why alcohol percentage increase near distillation? That we studied in respiration. You may be remembering it. That itself is a question, that value. Because yeast is the main organism utilized. So yeast poison themselves to death when the concentration of the alcohol reaches about 13%. That percentage itself is a question. So maximum up to 13% naturally we can get by using yeast natural fermentation. To increase the percentage of the alcohol content, then we have to distill. Then only we can increase the percentage of alcohol like brandy, rum, whiskey, etc., vodka, huge amount of alcohol content will be there. This is the reason why we have to distill after 13%. I think idea clear. It's a common question. Now, normally vodka from potato has got 60 to 80% of concentration of alcohol. While in beer, it is comparatively very less. Beer from barley malt concentration of alcohol is just 3 to 6%. Wine, it is slightly more than in the beer, that is 9 to 12%. Now, from the fruit juice, then whiskey from fermented cereals comes around 50%. Again, brandy from fruit juice or juices, 60 to 70%. Rum, again from molasses, 40%. Gin from rye malt, it is 40%. So based on substrate, alcohol content can change or product can change. I think idea clear that it can be a match the following type question. Even the NCERT not deals with it. Vodka, what is the percentage? Then beer, whiskey, rum, brandy, wine, what is the percent that way? NCERT no such a direct data give. If you know it is good. Now comes the term, another famous term from this chapter, antibiotics. So as usual, what is the meaning of the term antibiotics? Anti normally means against, biotic means connected with life. So antibiotic normally produced from microbes. This is considered as one of the most significant discoveries of the 20th century. So antibiotics against life. So against life, here it means against disease-causing organisms. It is pro-life with a reference to humans, because when infection comes, we'll take antibiotics, so the pathogens will be killed and we will be saved. That is, for us, it is pro-life. For microbes that causing diseases, for that, it is antibiotic. I think idea clear. Now, the term antibiotics, coined by famous scientist Selman Waksman, 1888-1973 lifespan, Nobel Prize in 1952. Now, the term antibiosis was given by Paul Williman, 1861-1932. And what is meant by antibiosis? It is the process by which life could be used to destroy life. That is, effect of antibiotic on microbes. That is antibiosis. Now, first antibiotic was penicillin by the famous scientist Alexander Fleming, connected with the famous bacteria Staphylococcus and the mold Penicillium notatum. That organism itself is a question. And the antibiotic is called penicillin. So when penicillin was there, bacteria can't grow. And remember, this penicillin discovery was a serendipity. Fleming was shocked to observe it, because those areas where penicillin was there, bacteria is not able to persist or live. Full potential of an effective antibiotic as an antibiotic, full potential, was established by another two famous scientists. Their names are, that is, Cheyenne and Florey. Full potential of the antibiotic. In World War II, this penicillin was extensively used to save life. And these three scientists, Fleming, Cheyenne, and Florey, together received Nobel Prize in 1945. Antibiotics sometimes refers to be wonder drugs because their effect and how they are able to kill the microbes and how they are able to save the life of humans. But remember, after regular consumption, an idea now came that is called antibiotic resistance. So going to be a headache for the human beings. Antibiotic resistance. Some diseases able to control by antibiotics include, that is NCERT 4 diseases, usual question. One is whooping cough, Kalikasi, by bacteria Bordetella pertussis. Diphtheria, Galgoto, by bacterium Corynebacterium diphtheria. Leprosy, commonly called Kushtrog, by bacteria Mycobacterium leprae. It is popularly called Hansen disease. And plague by bacteria Yersinia pestis, previously called Pasteurella pestis. So plague is a bacterial disease. Okay. Now next is chemicals, enzymes, and other bioactive molecules using microbes, and large number of chemicals has been produced, which include organic acids will be there, alcohols will be there, enzymes will be there, and even bioactive molecules will be there. So among these four chemicals, organic acid, alcohol, enzymes, bioactive molecules, many doesn't know what is meant by bioactive molecule. That is why I put that heading or question, what is meant by bioactive molecule, because we should know what we are going to study. Then only we'll be able to follow it easily, and that will be in our memory also. So what is meant by bioactive molecules? See, bioactive molecules are chemicals that interact with the living organisms and can have a positive or negative effect on them. It can be seen in almost all living organisms. They may work physiologically, biochemically, and they may even produce biological effect also. That is how we can define the term bioactive molecules. And under which certain chemicals we have to proceed. Some organic acid. So database question again. Match the following. Fungus, citric acid. Name of the fungus is Aspergillus niger, used in many areas like medicine, food preservation, rice, flavoring, etc. Second, bacteria. Chemical is acetic acid. Name of the bacteria is Acetobacter aceti, used for vinegar production. Third is again bacteria. Chemical is butyric acid, Clostridium butylicum, connected with the water. Now, fourth is bacteria, lactic acid, Lactobacillus, connected with curd. We already studied in first episode. Fungus, gluconic acid, Aspergillus niger, penicillium, source of calcium. This way, many applications are there. And different species, genus also available. These are the main items. Now, next is alcohols. Mainly fungus. Ethyl alcohol. Name of the fungus you are familiar, Saccharomyces cerevisiae yeast, unicellular eukaryotic organism, commercially used. Now, enzymes, a set of enzymes. Again, match the following popular question. Various enzymes and related chemicals are utilized in medical and industrial sectors. One by one. Starting with the first item, fungus. Name of the fungus is Candida lipolytica. Now, another name is the Yarovia lipolytica. Produces a chemical or enzyme lipases. Lipid digesting, that is how the term. Helps to remove oily stains from laundry. Another fungus, Geotrichum candidum, also can be utilized. Next is fungus Aspergillus niger. Similarly, Bisoclamis species. Connected with the pectinase, helping in a process called clarification of fruit juices, making them clear. So we should know, just like bioactive molecule, what is meant by clarification theory in its exam question. Clarification means removal of substances that makes juice cloudy, like pectin, cellulose, starch, protein, and metal. So after this clarification, content will be just like a watery event, smoothly flowing. That is called clarification. Now, next is, again, fungus and bacteria involved. Aspergillus, Motirella species, Bacillus. First two, fungus. And they are connected with the production of a enzyme proteases, again involved in clarification, like pectinase. Now, bacteria, Streptococcus. Enzyme is streptokinase. After modification, used as clot buster for removing clots from blood vessels of patients undergone myocardial infarction leading to the heart attack. So, clot buster, very famous chemical, streptokinase from Streptococcus. Now, fungus and bacteria, the names are Aspergillus rhizopus, species of Bacillus involved in production of amylase that will degrade starch. So, again, an important area of question, exam question, match the following directive, fill in the blank, different manner. Now, another fungus, another important one, fungus Trichoderma polysporum. Chemical obtained is cyclosporin A. And what is the cyclosporin A? It is an immunosuppressive agent. Where? In organ transplant patients. Main headache is when organs are transplanted from one individual to other. They will match it, but still there can be rejection. So to prevent it, chemicals like cyclosporin A is used as an immunosuppressive agent in organ transplant patients. And what is this cyclosporin A or chemically? It is a cyclic peptide having 11 amino acids. Now comes next microfungus yeast, Monascus purpureus, that is a type of yeast, which will be concerned with the production of a chemical called statin. Statin is a blood cholesterol lowering agent. And what is the action of it? That's another question. It is a type of competitive inhibition of the enzyme responsible for synthesis of cholesterol. Statin resembles another chemical called mevalonate and is competitor inhibitor of beta-hydroxy, beta-methyl, glutaryl coenzyme reductase, otherwise HMG-CoA reductase. So all famous, important for exams. And after that, you can find 5 theory questions. 5 NEET model question with the key. So, what all things we learned in this episode? Microbes in human welfare, second part. So, we can just rewind it. So, it will be easy for you, sorry. So, we started with an introduction, what all things we'll be learning, specifically connection with the different industries. Then, fermenters, fermentation plan, NCERT diagram, NCERT-based question, big vessels, large vessel. Then, fermented beverages. First, we define what is beverages. And then fermented beverages, main organism involved is yeast. And the products like wine, beer, whiskey, brandy, rum. Then Saccharomyces cerevisiae, bread making also it is connected. Reverse yeast here. Then for fermenting malted cereals, fruit juices production, ethanol production, all this. Yeast is involved. And depending upon the type of the raw material and type of processing, with or without distillation, different types of alcoholic drinks are obtained. Without distillation, that is a very famous question, odd one, wine, beer. With the distillation, whiskey, brandy, rum. Intoxication will be more. And main materials for ethyl alcohol production is molasses, fruit juices, malted cereals by fermentation. Common one is molasses. Alcohol percentage can be increased by distillation. What it means? Because yeast, when naturally fermented, it can produce alcohol up to 13%. Beyond that, yeast will sometimes or it will die. So maximum up to 13% alcohol content you can get when we ferment naturally. Some drinks has got 50%, 60%, even 80% alcohol. So we can increase that alcohol content. It is through a procedure called distillation. And different beverages with a percentage of concentration of alcohol. It's again famous question, match the following type question. Then we entered into antibiotics, meaning using microbes, wonder drug of 20th century, that way we can say. Then against life, that is for microbes. For humans, it is pro-life. Then 3 famous scientists concerned with antibiotic, antibiosis, its effect, etc. Salman Weizmann, antibiotic term, developed in 1952. Term antibiosis was given by Paul Williamin. Antibiosis means action of antibiotic, that is, process by which life could be used to destroy life. Then the famous scientist, first antibiotic penicillin by Alexander Fleming, connected with the microbe Staphylococcus and mold, Penicillium notatum. Chemically is penicillin, so bacteria cannot survive on it. And full potential as an effective antibiotic established by Cheyenne and Flore, 3 scientists. And remember, penicillin discovery was a serendipity, and Cheyenne, Flore. And in World War II, this penicillin was widely used to save the life of wounded soldiers. And these 3, Fleming, Cheyenne, and Flore, received Nobel Prize in 1945. Then, now you can find a new idea, new physiological effect came antibiotic resistance. Now, some diseases able to control by antibiotic includes, that is, whooping cough, diphtheria, leprosy, plague, their scientific name, and leprosy, that is otherwise called Hansen disease. After that, chemicals, enzyme, bioactive molecules in connection with the microbes. So, first we define or identify what is meant by bioactive molecule, chemicals that interact with a living organism and can have a positive or negative effect on them. It is present in almost all living organisms. Work physiologically, biochemically, may produce even biological effect. Now, after that, a set of chemicals, again, match the following important organisms, chemicals, how it is produced, etc. Then comes alcohol, same manner. Then enzymes, under which large number of enzymes, again, very important, for example, point of view, match the following, etc. Very important. So, as such a microbes, we think it is an easy chapter, but for question-wise, some important areas are there, NEET or theory. So, don't discard this chapter. 2-3 questions, sometimes you may get it. It's easy to get them. Then after that, we entered into 5 theory questions, 5 NEET model question with the key. If you like this episode, subscribe all, like it, share it, comment on it. And remember, I will be supplying a NEET model question with the discussion live and solution, etc. It will be gradually coming. So please subscribe the channel so it will be automatically coming in your channel, Botany. So thank you. We can meet later. Take care. Bye. All the best.